This UCSD TV program is a presentation of University of California Television for educational and non-commercial use only.
We all strive to live a long, prosperous, and healthy life. With advances in health and medical sciences, this goal is ever more attainable. The Sam and Rose Stein Institute for Research on Aging is a nonprofit organized research unit under the auspices of the University of California at San Diego, committed to advancing lifelong health and independence through research, education, and patient care. To better empower and improve the lives of young and old alike, the Stein Institute presents the following program.
I know that most of the people in the audience are quite young, and that's a really good thing. But someday you're going to get old. And so what we're going to talk about in the next little while is the brain and what happens to the brain during aging. And we won't get into great detail into the nitty-gritty of this, but I do want to say that surely at this time in our medical development, one of the greatest concerns we have as a population, as individuals, is what will happen to our memory and our cognition. Will it persist at a high level? How can I be as effective as possible in encouraging good old age with healthy cognition? I won't have any magic bullets for you, but I do want to tell you what we're up against and the kinds of things we're going to be seeing. So what I'll talk about is Alzheimer's disease as a dementing disorder, typically in the elderly. I'll tell you that not every memory problem is Alzheimer's disease. I'll talk about the incidence of Alzheimer's disease and why it in fact is an oncoming epidemic. We'll talk about that. I'll talk about some of the pathology that occurs to cause or is associated with Alzheimer's disease. Some of these changes in the brain, as well as genetic evidence, points to a number of possible disease mechanisms. We need to understand those disease mechanisms if we are to come up with effective treatments. And so we'll touch on those topics. And some of our own work that suggests one interesting approach to treating or even preventing Alzheimer's disease. So here are some questions. What is Alzheimer's disease? Why are so many people concerned about it? Why is it viewed as an oncoming epidemic? What do we know about pathogenesis, and what new treatments can we envision? So let's go in order. First, we have to define some terms. So dementia is a decline of memory and other cognitive functions sufficient to interfere with usual activities. Neurodegeneration is a disease state in which there's dysfunction and death of neurons that causes neurological disability. Now, I want to introduce two important biological terms here. One is neuron and one is synapse. And here's all the neuroanatomy you probably really need to know. The nervous system is the most exquisite, evolutionarily derived machine for processing information. That's what it does. It brings in information from another source, it processes that information, and it results in a change in function. Information received, processed, and sent. The wiring that makes that possible is composed of neurons. And so you see here neuron 1 and 2 and 3. And these are three neurons linked to each other in a circuit, a simple circuit. And each neuron has three very important parts. There's a cell body that has the nucleus, all the DNA is in there. There's an antenna that we call the dendrite. We call it dendrite because it has a tooth-like appearance, at least in cartoons. And then there's this long wire called the axon. And so what happens in the normal neural circuit is that information comes in to stimulate this dendrite. There's an electrical signal sent down the dendrite to the cell body. And if it's sufficiently large, it causes an electrical impulse to fly from the edge of the axon here all the way down the axon to this point called the synapse, this point where two cells come very close together. At this point, that electrical signal elicits the release of small molecules, which then diffuse across the synapse to excite their receptors, little receptor bodies, little acceptor sites on the dendrite of the second neuron. It's then stimulated, and the beat goes on as one sends information down the circuit. The processing comes from different neurons doing different kinds of things with different kinds of information. So this is the basis of everything the brain does. Everything you feel and think and love and hate, and all the tastes that you have, all the beautiful visions that you have, the beautiful music you hear, all of this is possible only because of the operation of neuronal circuits. It follows that we really need to know a lot about neurons and synapses, and that's our job in the laboratory and in the clinic. But it also follows that all neurological disorders are due to disrupted circuits. So here's the reality. Whether it's stroke or multiple sclerosis or Huntington's disease or Lou Gehrig's disease or Alzheimer's disease, every neurological disorder is due in some way or other to breaking the circuit or mini-circuits. In Alzheimer's disease, the circuits that are most prominently affected are those that control memory and learning and higher order function. Not all the circuits are affected in Alzheimer's disease, but those that are affected cause the symptoms that we see. Well, what about elderly people? What do they say? What do they complain about? This is a slide that was several slides donated to me by my colleague, Mike Raffi, who's a real expert in memory disorders and is very keenly involved in our memory disorders clinic here at UCSD. Older people have problems recalling names and words, recalling where they put things, knowing whether you've told someone something, forgetting a task after starting it, or losing the thread of conversation. Now, I know that none of you in this room has had any problem like this. But imagine, this is real data. You know, I think I can speak for myself in telling you that names are harder to remember now than they were when I was younger, let's say. So these are complaints, but these are not unusual complaints. These are healthy elderly. These are not people that have Alzheimer's disease. What patients say that have Alzheimer's disease and what people report about them are trouble with new memories, relying on memory helpers, difficulty finding words, struggling to complete familiar tasks, confusion about time and place or people, misplacing familiar objects, a new onset of depression or irritability, making bad decisions, changes in personality, loss of interest in important responsibilities, seeing or hearing things on occasion, and expressing false beliefs. A different level of severity and a longer list. So we can think of normal aging here as basically lasting a very long time, no problem with this, but in some normal aging results in something we call mild cognitive impairment. That's a person that has some problems with memory, but generally is functioning quite well. Those folks can go on either to have dementia or stable memory, or some of them actually show memory improvement. So what's mild cognitive impairment? In this case, people have complaints of memory. They say, I don't remember X, Y, Z as well as I used to. They, in fact, have test evidence, objective evidence of problems with memory relative to those of their same age or educational level, but largely have normal cognitive function. So that's mild cognitive impairment. What causes it? Well, it's important to note that there are a lot of things that cause mild cognitive impairment. One can have depression, and that may well improve with treatment of depression. Medical illnesses like hypothyroidism can be at play. There can have been head injuries that affect a person's ability to remember. Stroke can affect memory. And then there are degenerative processes of the kind we're going to talk about tonight, including Alzheimer's disease. Here's a slide that shows you on this axis the number of people that don't have dementia versus years of follow-up. So if at time 0 you collect a large number of people and you do careful cognitive testing, some of those people will remain at a very high level of function. In fact, only 1 or 2% of those people per year will go on to have dementia, this more serious involvement of memory and other cognitive functions. But people with MCI, those who had subjective complaints and objective evidence of memory loss, convert at a much higher rate, up to 10 to 15% per year. So MCI is a warning sign, is an early monitor of trouble going on in the brain. It may be transient and go away. It may be stable, or as shown here, it may convert to dementia. So let's go back to our list. What is Alzheimer's disease and why are so many concerned about it? Let's think about that for a bit. And let me show you some data. So this is the prevalence of dementia across a number of studies. Here's the number of those people who are demented versus age. And notice down here at age 61 to 64 is a very low number of people that would be able to be diagnosed with Alzheimer's disease. Now, this is a normal population. This is not a population in which one's identified genes that cause Alzheimer's disease. It's a pretty normal population of people, typical people. But notice that as age goes forward, the number of those people, the prevalence of those people becomes quite high. And in fact, there are estimates that at age 85 and somewhat beyond, as many as 40 or even 50% of the population are demented. Most of those people will be shown to have Alzheimer's disease. At least 24 over 36% will be shown to have that. So where are we now? Well, right now it's estimated there's about 5 million Americans with Alzheimer's disease. And that's a large number of people, mostly diagnosed, but not all diagnosed. So here's the number that are diagnosed and treated. So diagnosed and treated, and then here are the categories they fall into: mild, moderate, or severe. Most are 65 years of age or older, in agreement with that prior figure that I showed you, and a prevalence reaching 50% or more at age 85 and older. People with Alzheimer's disease typically live about 7 to 10 years after their diagnosis. I heard a discussion yesterday. The discussion looked at specifically at people that were 90 years old and older. And the goal was to decide what fraction of those people had dementia and what did not. A fairly large fraction had dementia, as you can imagine from this data. But they asked, in those old folks, 90 and older, that had normal cognition, did they convert to dementia or not? And if so, at what rate? And strikingly, they did convert to dementia at a rate of about 8% per year. Now, I asked the question of the speaker, is dementia inevitable if I live long enough? And they didn't want to kind of say that. And of course, there are the examples of the 122-year-old French woman who, smoked and drank and had fun and wasn't demented when she died of something else. But I think we can say reasonably that we're in trouble here if we think we're going to become very old without significant memory problems. Here we're looking at the oncoming epidemic in the USA. So here we are sitting right in here, and notice the marked increase in those that are getting older, and with that, the marked increase in those that are at risk for dementia. In the world today, there are about 36 million people with Alzheimer's disease. By 2050, there will be 115 million. Let's put that in perspective. if every one of these people has 2 caregivers, by 2050, almost 500 million people will be involved in having or caring for someone with Alzheimer's disease. We're talking about a population that's probably larger than the western half of Europe. An enormous number. An enormous number. So let me give you a little more data. So Alzheimer's disease is a dementing disorder of insidious onset with impairment in short and long-term memory. There's also trouble with abstract thinking and judgment. There may be a change in personality. Frequently there is. There's no disturbance of consciousness. These people are wide awake. And the disorder, as mentioned before, has its onset after 40 and most often after 65. In the early stage, symptoms come on and they're not too prominent, but they progress. There's increasing forgetfulness and less spontaneity and less insight. In the middle stage, memory's worse, and now there's frequent repetitions of questions and items are misplaced. Language skills are less good. There's trouble performing complex tasks. Calculation is poor. Finances become a problem. Getting lost becomes a problem. Wandering happens. Some are passive, some are restless, and there can be apathy or aggression. In the advanced stage, only fragments of memory remain. Verbal output is poor or nil, and it's very difficult for these people, or completely impossible for these people, to care for themselves. And they end up bedridden and mute, unresponsive, and they usually die with an infection of one sort or another. Not a happy prospect. Here's an artist, William Untermolen, a very excellent artist, and you can see these self-portraits that he put together over the course of his illness. So the first was painted just prior to diagnosis, and then you can see them wander down. And this is what happened at the end, 2000, not so long before his death, as I recall. An inexorable progression of the inability to make sense of the world, to receive, process, and send information. Now, I wanted to show you a movie. This is a movie that my predecessor, as chair of the department, made, an interview with a patient and a patient's wife. And I think you'll see here how it is that this disorder presents in a doctor's office.
Thank you for coming in today, Mrs. Harper.
You're very welcome.
Can you tell me how old your husband is?
He is 81.
Okay. And what age was he when he first developed his first symptom of this illness?
It was in 1995, so you have to do the arithmetic.
Okay.
Subtract
75.
Yeah.
Okay. And what was his first symptom?
Forgetfulness, not remembering events or names. It's hard to tell because a lot of people don't remember names. And then not remembering dates, losing things, couldn't find them, stuff like that.
Okay. And what has happened over this 5 to 6 year period of time?
The memory loss problems have increased. He's been in study programs, and I believe the trial medication has helped him because it has slowed it down. And but in the past month, he had surgery, and he was in the hospital for a couple of days and had anesthetic. And I believe that anesthetic moved his memory abilities down a notch.
Okay. And what are his main current symptoms?
Current symptoms are memory. He can't formulate sentences. This was not a formally educated man, but a well-read man who did a lot of writing and wrote poetry. And he could not do any of that. He used to read the newspaper every day. Now he can't. He'll say, he'll bring the paper to me and say, the world is so confusing that he can't figure it out what the newspaper is saying. Now he doesn't even try reading the paper.
Can he deal with making change or paying bills or handling money?
Paying bills, he hasn't done for several years. That was one of the early things to go. Making change, he did until about 2-3 years ago. But now he's in a situation where he emotionally, that's the biggest change. He seems to be totally dependent on me and doesn't carry money unless I insist he does. Okay. And so doesn't have much opportunity. I can't really measure that to see whether he still could do it.
Any difficulty with dressing himself?
Yeah, he needs constant cueing through dressing. Would put clothes on in the wrong order, and a shirt over a sweater, things like that. And fortunately, he's good-natured, and if I say he's got to change it, he will. Can't find anything, any of his clothes. I have to find it for him and lay it out, and that's been for a couple of years now, easily 2 years.
Okay, thank you very much. Mr. Harper, thank you for coming here today. Can you tell me whether or not you have a memory problem?
I don't think so.
Okay. So you think your memory is functioning perfectly okay?
Well, perfectly does not necessarily mean that I can repeat everything I want to say. But what's a perfect memory?
Okay, but overall you don't think there's a major problem with your memory?
I don't think so.
Okay, great. I'd just like to do a little bit of memory testing with you today. Can you
go right ahead.
Can you tell me what today's date is?
Today's date? Don't really know.
Okay, can you tell me the month?
The month. The month could be February or something like that.
Okay, and the year?
And the year... No, you better...
Okay, do you know the day of the week?
The day of the week, it could be Monday or Tuesday, but we won't be able to go to the movies because my wife wants to
Tell me which one it is. Is it Monday or is it Tuesday?
I would say Tuesday.
Okay, very good. It is Tuesday. And what season are we in?
Season would be the beginning of spring.
Okay, and what's the name of this hospital or medical center?
The name of this place?
Yes.
I always try to forget it as fast as possible.
Okay, and what floor of the building are you on?
What floor? It's probably second floor, maybe third floor.
Okay.
Because it depends how many.
Okay, and what city is this?
The city is San Diego.
And the county?
The county? I think it would be San Diego.
And the state?
And the state? That's a good one. State, no, I don't think I
Okay,
remember.
I'm going to give you 3 words. I'm going to ask you to repeat them after me and then remember the 3 words, and I'll ask you for them in a few minutes. The 3 words are ball, flag, and tree. Can you repeat that for me?
Ball, flag, and tree.
Very good. Okay, can you spell the word world backwards?
D R O W.
Okay, can you start with 100 and count backwards by 7s?
Okay, 100. Next would be 93, and keep going.
And keep going backwards by 7s.
93, going back. That's difficult.
Okay, let's leave that.
Yeah.
What were the 3 words I gave you a few minutes ago?
I think I forgot them already.
You think you forgot them already? Okay. What do you call this item that I'm holding up?
This is a watch.
And this item is?
A pencil.
Okay. Can you repeat after me? Just say no ifs, ands, or buts.
Okay. No ifs, ands, or buts.
Okay. Can you please take this paper in your right hand, fold it in half, and put it on the table?
This way and half or the other way?
Yes.
Doesn't matter?
Either way.
Okay, well, I'm going to fold it nice together, but hopefully nobody can read it. Okay.
Can you copy this figure on the piece of paper? I'll hold it for you.
Yes.
Good. Okay, and the last thing, can you draw a clock for me? Put in the numbers and set the hands to 3:30. Please note that Mr. Harper walks extremely well. He has a good arm swing, and he turns very nicely at the end of the length.
So I wonder if you saw what I saw there. What I saw was a very socially appropriate man who tried very hard to cooperate with Dr. Thal, but who had terrible problems with memory. He really was unable to remember things that I'm sure we all know and remember just automatically. The other thing that he did, which is very characteristic of patients in this stage, is he made light of some of his weaknesses or would deflect interest to some other topic. And you were laughing when he was laughing because it was kind of funny. But you know, I always try to forget the name of this place as soon as I get out of here. It's funny, but truthfully, it's an adaptation. It's a way of feeling better about himself, but also a little bit steering the conversation away from his weaknesses. Where are we in 2011 with treatments? I told you that we're concerned about ourselves, about our brain's ability to make sense of the world, and right now we're not very far. We don't have treatments that are very effective or that last in their effectiveness very long. So we have this class of compounds called cholinesterase inhibitors. They essentially raise the brain level of a neurotransmitter called acetylcholine. It makes sense on the basis of the pathology, but it doesn't work very well or for very long. There's another group of drugs, actually one drug called memantine, which works in an uncertain way, actually, but possibly by blocking a certain receptor, a neurochemical receptor. And it does seem to make a difference, but again, it's not changing the disease, it's treating the disease. It's symptomatic treatment, not disease reversing or disease stopping treatment. There are trials of vaccines against this peptide that I'll talk about called A-beta, and those trials are underway. We'll see if they produce some value. One is very hopeful for that. And then I should say there's several recent failed trials, and indeed there are far more failed trials in this disorder than there are successful trials. Let me show you a worrisome graph. So if we're looking here at the degree of a finding, and here at the clinical disease stage, I want you to note that memory function crosses a critical line out here, but that the fundamental pathology of the brain really begins quite a bit earlier. So if we're only making a diagnosis here, then we have a brain that's already quite severely affected by pathology. And so some would claim that the recent failures in clinical trials are due, at least in part, to the fact that we're trying to treat so late. What we really want to do is to move the curve over here so we can make that diagnosis as early as possible. So I argue that we understand too little the pathogenesis of Alzheimer's disease. Well, what do we know about pathogenesis? Question 4. What we can say is that there's a pathology of Alzheimer's disease that's quite informative. First of all, on gross examination at autopsy, the brain is quite atrophic. That means it lacks full volume. Here it's made pretty clear. What we've done is to take two brains and we've cut them in half and we put two different halves together. This is the healthy brain, and you'll notice that here's the matter. The brown staining is really the gray matter, and the white staining is white matter. The neurons live here, and their axons live here. You remember the two parts of the neuron we referred to. cell bodies and dendrites really here, so 2 parts up here, and then those long axons are sitting here in the white matter. And it's really a robust brain. It looks normal. Here's a space in the brain called the ventricle, and it's quite small. Look what happens with advanced Alzheimer's disease. The gray matter is less. You can see much thinner gray matter strips here. The white matter is less, and as a result, the ventricular space is much bigger. Notice here and here, one part particularly severely affected is the hippocampus, which is terribly important for short-term memory. So the brain is atrophied. One can pick this up on MR scans. So here is the cartoon of the type that I've really just shown you. Here are the normal brain, and here the Alzheimer's disease brain. And look here then on the normal scan and on the Alzheimer's disease scan. Again, lots of space between these reddish strips. That's the gray matter as colored on this false rendering of the MRI scan. So notice there's much less brain matter in that same skull space as a result of brain atrophy. One can now actually develop a pretty high degree of reliability and predictability of disease by doing these structural MRI scans. And so here's a particular person that was scanned here at UCSD, and notice that his left and right hippocampal volume is less than 2 standard deviations below the mean, and that the left and right lateral ventricle is well above 2 standard deviations greater than the mean. Both evidence of atrophy of the brain. What about the microscopic examination? And these are characteristic features of Alzheimer's disease. Plaques, tangles, loss of synapses, and loss of neurons. Here's a piece of cortex from a patient with Alzheimer's disease. Here's the surface of the brain. Here's that gray matter that I talked about before with the cell bodies and their dendrites. And then here's white matter in this darker stain, this dark brown stain. And this is not normal. And what I want to tell you here is that these dark puncta, these dark brown puncta here, are really pathological lesions. And at higher power, what they are plaques, spherical little accumulations that show the presence of both axons and dendrites, shown here in the dark, thin, wiry staining, and a peptide called the A-beta peptide, and we'll talk more about that. Quite abnormal. The tangles are made of a different protein called the tau protein. This tau protein is very important for keeping axons stable, helping the cell move cargoes around. And that becomes quite insoluble and quite abnormal in its structure. And you can see here cell body full of these tangles, and here are neurites, these either dendrites or axons that are full of this abnormal protein. Abnormal not because it's not normally made, but abnormal in this case because it's modified. It's changed in its composition as a result of phosphorylation, a chemical change that modifies its properties. You can use PET imaging to detect amyloid. So I talked to you about the plaque, the A-beta. It stains with a certain set of dyes that say it's an amyloid. It has a certain basic chemical structure. And using the right tools, one can actually see in the Alzheimer's disease patient, shown here in red, where that amyloid is accumulating. So here's a control subject. Here are 2 different patients with MCI. Remember, we talked about minimal cognitive impairment. Here's a 3rd patient with MCI, and here's an AD patient showing a lot of red staining. So these plaques, these amyloid imaging defects, really are showing you the A-beta peptide in plaques in the brain. They arise years before people develop memory loss. And about 30% of people, otherwise normal people, have above 70, older than 70, have positive scans. So we now have certain biomarkers that can help us detect and diagnose disease. So I've mentioned brain atrophy, and you can look at that with MRI studies looking at brain atrophy and regional atrophy, and certain new studies that are available that talk about how parts of the brain are connected. Or you can do functional studies, and here again one can look at the activation of the brain One can look at its glucose use, which is decreased specifically in people with Alzheimer's disease. And the biochemistry of spinal fluid is also useful. One sees changes in the peptide levels for A-beta 42 in this phosphorylated form of tau, and one can do additional special studies, including the PET study that I showed you just a slide or so ago, looking at amyloid deposition. Here's this glucose study where it's red is where neurons are active and metabolizing glucose. Here's the normal brain. Here's MCI, and look at the marked decrease in glucose use in the brain of the person with Alzheimer's disease. Neurons are lost, synapses are lost, and we can look at this in some detail. The point to make here is, to summarize this, is that the disorder follows a characteristic clinical course with a relatively uniform set of pathological changes, is largely sporadic, but, and very importantly, a significant number of cases are found in families or in another clearly genetic context. So they include early onset familial Alzheimer's disease, families in which there's an earlier onset of Alzheimer's disease, people with Down syndrome, and in cases of gene duplication, which is a kind of early onset familial Alzheimer's disease. And all of them, all of these disorders, implicate in one way or another the APP protein or its products. And let me show you a slide that makes that point. Oh, before I show you that, another piece of the pathology that's very, very characteristic and quite early is an enlargement of a cellular organelle called an endosome. Endosomes are pieces of surface membrane that are pinched off and brought inside the cell. And what's clear is that whether it's a person with Down syndrome who's older or a person with Alzheimer's disease, these endosomes are quite large. Now, you may know this, but let me say it clearly. Every person with Down syndrome shows all of the neuropathological features of Alzheimer's disease by age 40. And if they live long enough, we believe that every one of them will go on to have Alzheimer's disease-related dementia. And that's a very interesting fact, and we know something about their genetics. One piece of which is that of those 350 genes that they inherit, the 350 genes that come along with the extra copy of chromosome 21 is a copy of the gene for APP. This amyloid precursor protein, APP gene, is terribly important in the pathogenesis of Alzheimer's disease in people with Down syndrome. So what's this all about? Well, we know that the proteins that regulate these endosomes are increased in the brains of these subjects, that an increase in one of those proteins in particular called RAB5 correlates with cognitive decline, that the increase in the number of these RAB5 positive endosomes are early, and that one can begin to think about linking this pathology and the normal function of the endosome, which in part is to carry trophic signals between neurons. So I showed you the picture in which one neuron spoke to the other neuron using electricity and neurotransmitters, but the second neuron speaks to the first neuron also by releasing small molecules that bind to receptors on the axon and carry a trophic signal back to the cell to say, stay well, you've got a partner out there. So the genetics of AD looks a little like this right now. We know that there are mutations in the amyloid precursor protein, and in some families what seems to be abnormal is principally an extra copy of just this gene. We know there are mutations in proteins that metabolize APP called presenilin 1 and 2. In fact, there's more than 200 mutations in these genes. And we know that in Down syndrome, it's very likely an increased dose of the APP gene that plays a conspicuous role in causing Alzheimer's disease. More evidence for that is that there are people that we know about now who live to old age who have not lost cognition, one of whom has died and come to autopsy, no plaques and no tangles. And what's abnormal about their genome, otherwise look exactly like anybody else with Down syndrome and function that way. What's different about them, what is correlated with them not having Alzheimer's disease, is only 2 copies of the gene for APP. They lost the other copy somehow. So very powerful evidence that Down syndrome is predicated in its ultimate predilection to Alzheimer's disease, at least in part by an extra copy of the gene for APP. Now this is the protein APP. It's a pretty big protein. Here we're showing 770 amino acids. And I want to point right here to this little orange square. That's where this A-beta peptide resides. And in fact, you only get to that peptide as a result of cutting this bigger protein in several pieces. And what's really interesting from the genetics is that, and you see the arrows here, all of the changes, all the mutations that are linked to Alzheimer's disease are in this very region where the peptide resides. And so this tells us that this peptide is almost certainly going to play a role, its processing play a role in the genesis of Alzheimer's disease. Pretty strong conclusion, but I think a very reasonable conclusion. Something about the structure of that peptide is quite significant, and its processing is quite significant. Now, how is it processed? Well, it turns out in the endosome, this protein, now we have a picture for APP, gets in the endosome and is processed by presenilin 1 and 3 other proteins in something called the gamma secretase complex. And as a result of that, one releases a fragment that goes into the cell, one releases the A-beta peptide, which pops out into the lumen of the endosome, and subsequently might be secreted. Outside the cell, it may form plaque. It does form plaques, and it may contribute to toxicity. But what's very characteristic in families with Alzheimer's disease, there's a little bit more, a little bit higher ratio of proteins that are 42 amino acids long versus 40. And that appears to be quite critical in allowing for the pathogenesis of this disorder to go forward. Other genetics that one should know about are ApoE4, which increases the risk of Alzheimer's disease but does not cause it itself, and a number of other genes that have come forward recently, which presumably also act as risk factors and probably only very rarely as actual disease-causing genes. There are just a raft of possible mechanisms that could cause Alzheimer's disease. So all the genes we've talked about and all the pathology we've talked about creates a long list of possibilities. And it's for the investigators to figure out, where should I go? How should I take this list apart? How can I possibly evaluate what's going on? So the players in the mix are APP and its mutations, mutations in presenilin, aging, plaques, tangles, big endosomes, various structural forms of A-beta that people think are more significant. How do you make sense of this whole thing? Well, there's a dominant hypothesis in the field that says that A-beta peptide is really critical. And it's supported by genetics data, biomarkers, neuropathology, disease models. And in clinical trials, those attempts to reduce the levels of that peptide have failed. And so that's why the question mark is there. And so the question comes again, are we treating too late, or do we need to think differently? Is there another way to think about the cause of this that might help us better understand it? Ultimately, I think the question might be asked, how is A-beta 42 toxic? So one is that it's the whole story. It is the toxic fragment. It causes all the misery by interacting with one or more proteins in the cell or on the surface of the cell or outside the cell to cause cells to become sick. And or, possibility two, A-beta 42 increases are due to some other problem in the cell. And we love the possibility that it simply marks a decrease in the activity of this enzyme, the gamma secretase complex. In other words, instead of chewing to shorter peptides, we think the enzyme stops too soon. And this A-beta 42 is one marker of that. But guess what? That same enzyme has 90 other substrates. So one could imagine that a deficiency of that enzyme is responsible more globally for changes in neurons that don't all depend on just A-beta. Here's what we think about. We know that endosome sizes are increased, and they're increased early. We know that big endosomes traffic poorly. They cause neurons to lose strength and vitality because they don't see their targets anymore, and that ultimately leads to neuronal death. So we think. We know that Down syndrome causes this and this. We know that Alzheimer's disease causes this and this. And the question is, how does A beta 42 fit in, if at all? What we've looked at then in our studies is the trophic deprivation idea. Could one explain the loss of cell viability and function by it not having sufficient trophic factor support? What are we talking about here? So here is this endosome that I talked about before, but importantly notice that this carries a trophic message. In essence, the trophic factor produced in this spine, this dendrite, is released and binds to a receptor, and that receptor is internalized with signaling proteins, and this whole complex comes back to the cell body to say to the cell, be well, be strong, you've got a target. Don't stop functioning. Now, here I've shown you the cell body and the axon and the synapse, yes? But I've really simplified this, because if we really looked at things, it would look like this. Here would be the cell body and the dendrites. Here would be the axon terminal, and this line going up and down is the axon. The axon is literally 1,000 times longer than the cell body is wide. And so now you see the real problem that neurons have. Neurons with long axons have to ship all their goodies, all the things that make synapses work, all the way through this network of axons. It's one axon, but it goes back. Obviously, it's one axon. It just goes very long distances. It's got to go that entire distance to get stuff from the cell body out here, and importantly, it has to pick up these trophic messages here and carry them all the way down the root until it gets to the cell body. Here's an example of what that really looks like. If you had a beach ball and you said, that's my cell body, and you put it at the Arc de Triomphe, hopefully you've been to Paris, then the axon attached to that beach ball would be less than 1 inch across and would run all the way from the Arc de Triomphe down to the Place de l'Encore. That's a long ways to send important information. So we really believe that the axon is highly vulnerable. Let me blow it up a little bit to scale it for you. If the endosome was the size of a car, it would travel through a tube 60 feet in diameter at 250 miles per hour on an undulating roadway, confronting a multitude of obstacles, some of which are also going 250 miles per hour in the opposite direction, and using as a source of fuel something we call mitochondria that are running around in that axon too. They don't stay stable, they run back and forth. I mean, think about how difficult this is to organize. So we think axons are vulnerable because they're so long, because they're so complex at the molecule level, and because the demands on them are so great. So how do we study this process of trophic factors moving in axons? And what we've done is to take one or more trophic factors and we've added a little chemical called biotin. Then we've attached this little dot called the quantum dot. So here's the molecule, here's the quantum dot, and with that, they're so bright, these quantum dots, that we can see them move in real time in axons, individual axons. And we use special chambers so that we put all the cell bodies here and all their axons grow out here. That's the top view, this is the side view. It doesn't leak at all, it's very handy. And then we can look right here at this, in this little space, and we can watch traffic happen. So here's a movie of a trophic factor moving in axons. These are hippocampal neurons. The trophic factor is called BDNF, and I'll show you what it looks like. Every white dot is an endosome containing one molecule of BDNF. And so they move, and they pause, and they move again, and they pause, and then they move. And with the system, one can readily map the traffic of literally thousands of endosomes. So the data that one collects is enormously detailed. You can see them move, and they pause, and they move. Here's one axon with 10 or 15 different endosomes moving down it, and one can see the very orderly behavior, move, pause, and move. We don't understand all the details of this, but we can measure with enormous precision. So could a failure of trophic traffic signals A failure of trafficking those signals contribute to neurodegeneration in Alzheimer's disease. Here's some evidence in support of that. Whereas there are decreased levels of NGF in the cell bodies, there's too much in the target where it's being made. But there's no change in the level of RNA, so the increase in the target must be due to the stuff in the target just sitting there. We know there's defective retrograde transport of NGF in mouse models of Down syndrome and Alzheimer's disease. We link these defects to degeneration of neurons, and the degeneration we see is directly linked to APP gene dose. Now, we study mouse models of Down syndrome, and we do that because, as I mentioned, everyone with Down syndrome gets Alzheimer's disease, and effective mouse models should model the degeneration of those neurons. Here's a little mouse that we use, an extremely effective model. Here's the little guy sitting here. He's got 104 genes, mouse genes, that are homologous to those on human 21. And in this mouse, we can actually ask the question, what about endosomes, and how do they move? Here we're looking at the hippocampus, and what we can say here is that these endosomes, just as in patients, are too big. If we remove one copy of the gene for APP, but all the other genes are there, we go from almost no transport to more than half normal transport, and we completely prevent the atrophy of these little neurons called cholinergic neurons. And that's also true with another population of neurons that dies in Alzheimer's disease, the neurons of the locus coeruleus. And so what we can say with some confidence is APP gene dose matters. And what we can say with additional confidence is that APP gene dose is linked to the size of endosomes being too large. So in this culture, from the mouse model, endosomes are enlarged. One can see that on this figure, where the relative number of endosomes is very much different between 2N, the normal mouse, and the TS65DN mouse here in the largest size class. And one then can compare the movies that one takes for the normal neuron and normal axon and the Down syndrome model axon right here. So the top two panels show you the movement over time of an endosome. It moves, it pauses, and moves. It moves, it pauses, moves, a little pause, and move on. Look at the marked difference here. One sees that the movement speed is about the same, but the pauses are more frequent and they're much longer, such that the net rate of movement here is only 1/4 the rate of movement in the normal axon. I won't describe all the data that supports this, but what we can say with some confidence is that an increase in these partially processed APP protein molecules causes an increase in the activity of that enzyme that makes endosomes big. We believe the same is the case for A-beta-42, this product from familial Alzheimer's disease as well as sporadic Alzheimer's disease. And we believe this cascade is an important part of pathogenesis. So we believe that, yes, A-beta plays a role, but it's very possible that other things are playing an important role in pathogenesis. And I specifically refer to the gamma secretase story to say, wouldn't it be exciting if that was the case? Because perhaps we can find a way to make that enzyme more effective. Can we do it? Well, in fact, we now have, through work at UCSD, just discovered a class of compounds that seems to make that enzyme work better. So let me show you very briefly these data. These are the bad guys in the red trace and in the purple trace. Those are the bad guys. That's A-beta-42 and A-beta-40. What this compound does seems to enhance the activity of the gamma secretase. So now those peptides go away, and instead much shorter peptides come along. And we guess that that is going to make a big difference in the size of endosomes, shrinking them to normal, decreasing the activity of this enzyme that's too active, and hopefully freeing up trophic traffic, and possibly increasing activity against all those other 90 substrates that this enzyme acts upon. So I'll summarize briefly. Alzheimer's disease is a dementing disorder. It's typically occurring in the elderly. It may be inevitable if we live long enough, unless new things happen. It's important to rule out other conditions for people with memory problems. The incidence of AD qualifies it as an oncoming epidemic. The brains of people with AD show characteristic features. These changes and new genetic evidence are really helping us to understand what might be going on. We need effective treatments. We sorely need them. And I'm optimistic that the additional research that is going on now and will go on in the future is going to point to those treatments and really rescue all of us from what is an unsavory end to our lives. I want to thank the fellows and the gals that work with me in my laboratory: Pavel Belachenko, Alexander Klishevnikov, Ahmad Salehi, Mike Maloney, and Xingbiao Wu. Important contributions also from Rachel Noshani, Steve Wagner, who's the chemist and the biologist who discovered this new set of compounds here at UCSD, and April Weissmiller. And these are the folks that support us, and we thank very much the National Institutes of Health, the Hillblum Foundation, the Thrasher Foundation, the Alzheimer's Association, Cure Alzheimer's Fund, and our good friends at the Down Syndrome Research and Treatment Foundation. So thank you very much for your attention.